Hello everyone, my name is Sebastián Urquiza. I am a PhD student in biochemistry in Chile, and now I will explain my poster entitled Epitranscriptomic m6A changes in coding and non-coding transcript induced by hypertrophic stimulus in the neonatal ventricular rat myocytes. This work is a collaboration with researchers in Brazil, and we are interested in our lab to studying the topological RNA modification in long non-codings, in discover novel long non-codings, and to studying in which biological process these long non-codings could be involved. So first describing our model, that is cardiac pathological hypertrophy. That's a condition that when the neurohumoral changes of the heart, we can have the reprogrammation of fetal gene expression. And basically here we have like the norepinephrine, like it's a catecholamine, that we have this receptor or cell signaling cascade. And in general, we have like this translocation of M-fat base of calcineurin that is a phosphorylation of M-fat. So the fats move on to the nucleus and restart the fetal reprogram to induce the expression of pro-hypertrophic markers that is a irreversible phenotype. And here in our lab, we are like really interested in studying the long non-coding, specifically in the hypertrophic context. But in several studies, we have discovered that these long non-codings could be modified, and the modification can alter the function or the native function of the long non-codings, like RNA processing and splicing, specifically microRNA sponging, like acting like a competing endogenous RNA network. And in general, also we have found that RNA stability and decay is also involved in this RNA modification context. But we are really interested in that our model is like in a norepinephrine cardio myocyte hypertrophy context, and how these long non-codings are novel non-codings based on the rat genome that is not well annotated. This non-coding transcript will be involved in this. So for move on, we have implemented a wet lab strategy and a dry lab, basically of based on the bioinformatic, tube informatic approach that I will continue explaining. But first of all, we have neonatal rats of the strains Sprague-Dawley, and we have like a primary culture of the ventricular myocytes of these neonatal rats. And we have two conditions, the control one and the hypertrophy one. We stimulated cardiomyocytes with norepinephrine at 10 micromolar, and for the RNA extraction, we take 3 samples, 3 ends of each condition, and then we have like pulled this condition to make the RNA sequencing with a mini INO box or nanopore technology. And the raw data of this analysis, we have implemented two different approach. It's a transcript discovery pipeline and m6A identification pipeline based on different tools for describing and to characterize this. And here we have like RNA classification. We have different classes of long non-codings, like intergenic long non-codings, gene, and that they were in between 2 genes, and in antisense long non-codings, and also antisense intronic long non-codings. And using RNA mining tool, we have like describing to separate the non-coding potential gene, and then the classification as well. Well, what we found here is like we also discover novel coding transcript, not only long coding, and based on the ensemble that were long codings that were reported in this annotated GTF. Also, we have discovered novel ones of the SOM classification based on the length of the exons. Here on the classification of mono multi-exons and the difference length of the RNAs, the new ones and the simpler ones reported as well as the long codings. And what we found here is the specific cardiomyocyte pro-hypertrophic markers like MPPA, ACT1, and MPPB that were like they almost classified as the pro-hypertrophic markers that we also validate by qPCR, but here in the RNA sequencing we also found it that were really upregulated in between the control and hypertrophy condition. And for the long colonies that were most expressed here, we have found that there were different reported long colonies as well as the ones that were novel, because here is the label of the string type put of the novel transcript that they discovered, and there was some of these novel and conservative long colonies that were upregulated in hypertrophy, and someone is like downregulated. But here also remember that we don't have too much samples, so we pull them so we can make a statistical analysis or differential pressure analysis. So only we have counts per million analysis. And to look up for some example of these genes, sorry for these long non-codings, we took the list and the ID of this m6A modification and input in a genome browser, the UCSC genome browser, and all this was filtered by only long encodings and the ones that were hyper or hypomethylated with m6A. And here we can look some examples. Here is like an example of intergenic long encodings. Is upstream this gene is EMC10, and if you look up, there's a coverage of the sequences that is like mapping here not only the m6A modification based on this genome browser, but here is like discovering or trying to annotate these novel ones. And also we have like putting the August ab initio gene prediction to look up the coding genes that were in the genome browser to check out. And here, well, the interesting thing is that in these two examples of the novel long non-codings that we found, hyper or hypomethylated, there is some marks that were lost in the two conditions. Here we have like a hypermethylated compared these regions, so we're interested in look up in the future why this methylated region is like hypermethylated or hypomethylated in a treatment with norepinephrine. So we try to look up which was the distribution of these and the frequency of these m6A modification as well. And we look here like there is like a common modification for coding genes, and there's like two is more representative for the control one based on the norepinephrine context one. So in both sides of the coding and non-coding genes, we found more m6A mark and frequency in the control based on the norepinephrine induced cardiac hypertrophy. So we're interested in how and which genes are hypomethylated, or they are like derived to decay, or even in the downregulation ones. And in the topological context, we don't know. We only describing the number and percentage of m6A based on this expression. And for final highlights or the conclusion of this work, preliminary results is that we found differential expressed transcript, specifically long non-codings in these neonatal ventricular rat myocytes induced by norepinephrine, using two approaches. Is related to reconstruction a transcript approach of long read sequence based on nanopore Oxford nanopore technology, coupled with m6A modification pipeline. And there is a differential methylation of coding transcript and long non-coding between both conditions, presenting a decrease in m6A modification in hypertrophy induced by norepinephrine. And this is the first study that described long non-codings and hyper or hypomethylated in a catecholamine-induced model in vitro. Thank you, and I am really very interested in having your feedback. And if you have in person, or also we can meet on Montreal and have to discuss. Thank you very much.